You know that feeling when you haven't had a good conversation or connected with someone you care about for a bit?
Hmm.
That little tug of unease, maybe even a sense of something missing.
Yeah, absolutely.
It turns out that's not just like a fleeting emotion. It's wired into us. Really fundamental.
It is, almost like hunger or thirst.
Exactly. And that's what we're diving into today. This feeling, it seems, has really deep biological roots.
That's a really important way to frame it, actually. We often think of social connection as, a bonus, something nice to have.
Right?
But this new research, it's really pointing to it being a core biological imperative, right alongside those absolute survival basics.
And the science is catching up.
Yeah, the fascinating part is scientists are now starting to actually map the specific brain circuits that drive this need. It's quite detailed.
Okay, let's unpack this then. We've got a really interesting piece of scientific research as our source today.
Recent paper, yeah.
And it takes a very close look at this using mice, believe it or not.
Animal models.
Now, we always need to be careful, right? What happens in mice isn't always a direct copy-paste to humans.
Definitely not. But the fundamental processes,
Yeah.
the basic drives, they're often conserved across evolution.
So these findings probably give us some pretty profound clues about ourselves.
Absolutely. I mean, we can't ethically do this level of detailed brain investigation in humans in the same way.
Right.
So the fact that these drives seem so basic suggests these mouse findings are incredibly informative. They tell us a lot about how our own brains might manage our social requirements.
So what's the mission here? What are we trying to understand in this deep dive?
Well, think of your social life maybe as having its own internal regulator.
What the thermostat?
Exactly, like a thermostat. And this research is really digging into the brain's social thermostat. Scientists are calling this process social homeostasis.
Social homeostasis.
Yeah. And they've pinpointed specific brain areas involved. And they even stumbled upon a surprising role for something we often overlook.
Which is?
Touch. Physical touch.
Interesting. Okay, we'll definitely get to that. But first, how do they measure this social need?
Right. So a key indicator that this social thermostat is working is something called social rebound.
Social rebound.
Yeah. After a period of being alone, animals, and we see this in people too, they tend to engage in much more intense social interactions when they get back together.
Like they're making up for lost time.
Exactly. It's as if their system is trying to catch up on what it's been missing. It's a quantifiable behavior.
Okay. And in this study, the researchers actually measured this social rebound in different types of mice.
They did, after isolating them for different lengths of time. And here's where it gets really interesting.
Go on.
They discovered that some strains of mice, like these FVBNJ mice, were incredibly sensitive to being alone. They showed a really, really strong urge to reconnect. A big rebound.
And others?
Other strains, like the common C57BL6J, they were more moderately sensitive, still showed rebound, but not as intensely.
Wow. So the type of mouse mattered. That suggests genetics.
Exactly. This difference between the strains really hints that there's a genetic component to how strongly we, or at least mice, need social interaction.
Huh. But maybe those FVB mice are just generally more anxious or stressed. Could it just be general stress?
That's a great question, and they looked into that. What's fascinating is they found no link between the mice's overall stress sensitivity, how they reacted to other stressors, and the intensity of their social rebound.
Really? No link?
Nope. And that's a crucial insight, because it suggests that this need for social connection isn't just some side effect of general stress.
It's its own thing.
It appears to be a distinct, fundamental drive, all on its own.
All right, so the stage is set. A core social need. A way to measure it via rebound, hints of genetics. Now, the brain parts, where is this happening?
Okay, so the research really shines a spotlight on a specific area, the medial preoptic nucleus, or MPN.
MPN, where is that?
It's located deep in the hypothalamus. It's a region we already knew was involved in various social behaviors like parenting, mating, stuff like that.
Okay, so not a totally new player, but they looked closer.
Exactly. What's new and exciting here is how they looked. They managed to monitor the activity of individual brain cells, individual neurons, within the MPN.
While the mice were just living.
In freely moving mice, yeah.
Yeah.
Both when they were socially isolated and then when they were reunited with others. It involved a pretty sophisticated technique called microendoscopy calcium imaging.
Microendoscopy calcium imaging sounds incredibly precise.
It is. It lets you see which neurons are firing in real time.
So what did they see happening inside the MPN? What were the neurons doing?
This is the core finding, really. They discovered two distinct groups of neurons right there in the MPN. And they behaved in opposite ways, depending on the mouse's social situation.
Opposite ways? How so?
Well, one group they labeled MPN isolation neurons. Guess what they did?
Let me guess. Active during isolation.
Bingo. These neurons became active when the mice were alone, and then, remarkably, their activity just plummeted. It rapidly decreased as soon as the mice were reintroduced to their companions.
Wow. Like a switch flipping off.
Pretty much. It was almost as if these neurons were directly tracking the state of being socially isolated.
So the brain literally has loneliness neurons, in a sense.
You could sort of think of it that way, yeah. Signaling that state of need is like part of that social thermostat we talked about.
Okay, that's incredible. So if those are the loneliness neurons, what was the other group doing?
The other population they identified, they called them MPN reunion neurons, and they did the exact opposite.
So quiet during isolation.
Right. Relatively silent when the mouse was alone, and then bam. They suddenly became active when the mice were back in a social setting interacting.
Suggesting they signal.
What?
Social contact, the good part.
It strongly suggests they play a role in signaling the presence of social interaction, yeah?
Yeah.
Maybe the satiety signal.
And did the activity levels, like how much these neurons were firing, connect back to that social rebound behavior?
Yes, absolutely. They found that the intensity of activity in those MPN isolation neurons, the loneliness ones, directly correlated with the strength of the social rebound later.
So more firing during isolation.
Meant the mice sought out social contact more intensely when they got the chance. It really reinforces the idea that these particular neurons are signaling the level of social need.
It's like the longer you go without connection, the louder these neurons shout, driving you to seek it out.
That's a good analogy. And they use some clever analysis too, principal component analysis, PCA.
What did that show?
It showed that the overall pattern of neural activity in the MPN was distinctly different during isolation compared to reunion. You could statistically separate the two states just based on the neuron firing patterns.
And the switch was fast.
Very fast. The transition between these neural states happened really rapidly right at the moment of reunion.
Okay, but were these neurons just about being alone socially? Or could, say, being hungry turn them on? Or being stressed in a different way?
Crucial question. And they tested that. They exposed the mice to other kinds of stressors like food deprivation or being physically restrained.
Yeah.
And they observed that the MPN isolation and MPN reunion neurons didn't show a strong activation in response to these other types of stress.
Ah, so they're specialized.
It really highlights the specificity of this system. It underscores that these specific neuronal populations seem pretty dedicated to processing the social context, not just general stress or arousal.
Okay, let's circle back to those different mouse strains for a second. The FVB mice, the ones with the really strong rebound, and the C57s with the moderate one. Did they see differences in these MPN neurons between those strains?
They did indeed. It connects back nicely. While both strains had both types of neurons, the isolation and the reunion ones, the MPN isolation neurons showed a significantly stronger level of activity in those high rebound FVB mice after isolation compared to the C57 mice.
So the loneliness signal was stronger in the strain that felt the need more acutely.
Exactly. This difference in neural activity could very well be the explanation, or at least part of it, for why the FDB strain exhibited that more pronounced social rebound.
Okay, this is fitting together nicely. We know where this is happening, the MPN. We know there are these two opposing neuron types tracking isolation and reunion. But what kind of neurons are they? What's their, molecular identity?
Right. Good question. To figure that out, the researchers used some really advanced genetic tools. Very cool stuff.
Like what?
Well, one technique was called TRAP2. It basically allows them to put a genetic tag on neurons that have been recently active. So you isolate a mouse, its isolation neurons fire, and boom, they get tagged.
Clever.
And they combine this with another technique called in situ hybridization. This lets them see which specific genes are being expressed, turned on, inside those tagged active neurons.
So it's like molecular detective work at the cellular level.
Precisely. Find the active cells and see what makes them tick genetically.
So what did this detective work uncover about the MPN isolation neurons?
They found that the MPN isolation neurons are primarily glutamatergic.
Meaning they use glutamate, the excitatory neurotransmitter.
Exactly. They use glutamate to send their signals. They also identified a specific set of genes that were particularly abundant or enriched in these neurons, genes with names like MP4R and CARTIPT.
Okay.
And these genetic markers matched up perfectly with a specific cluster of neurons, cluster E8, that the researchers had previously mapped out in that brain region based on gene expression.
Got it. And the MPN reunion neurons, the ones active during social contact.
Those turned out to be mostly GABAergic.
Using GABA, the main inhibitory neurotransmitter.
Correct. So they primarily use GABA to send their signals, which usually dampens activity in the neurons they connect to. They were enriched in a different gene marker, TAAR, and corresponded to a different cluster, I11.
So we have these two distinct populations, excitatory loneliness neurons and inhibitory reunion neurons. That's a pretty elegant push-pull system.
It really seems like it. And they even did a bit more digging on one of those genes you mentioned, MC4R, which was high in the acylation neurons, right?
Yeah, what about MT4R?
They specifically looked at MPN neurons that express the MC4 gene. Using other techniques, they confirmed that, yes, the majority of these MPN, MC4 plus neurons were indeed activated by social isolation and then inhibited by social reunion.
Just like the broader MPN isolation group.
Exactly. It helped solidify the identity of that population. And importantly, they checked, again, these MC4R neurons didn't activate strongly in response to other contuitive behaviors, specific to social isolation.
Okay, so we know these neurons exist. We know roughly what kind they are. But the big question is, what do they do? If we mess with them, does it actually change how the mice feel or act socially? What's their functional role?
All right. This is where the research gets really compelling, I think. They moved on to manipulating these neurons directly.
How?
Using optogenetics. This is that amazing technology where you can use light to turn specific genetically targeted neurons on or off.
Like a light switch for brain cells.
Pretty much. So first, they took mice that were already living in groups, socially satisfied mice, you'd assume.
Okay, not lonely.
Right. And they artificially activated their MPN isolation neurons using the light.
Turned on the loneliness neurons in socially happy mice. What happened?
It actually triggered social interaction. These mice started seeking out others more and even showed an increased preference for being around other mice.
Whoa. So even when they weren't actually deprived, turning on these specific neurons made them act like they needed social contact.
Exactly. And get this even more remarkably, this activation also led to a subsequent social rebound.
After the light stimulation stopped.
Yes. Even after they stopped artificially activating the neurons, the mice later showed increased social seeking. This suggests activating these neurons doesn't just cause a temporary urge, it has a more lasting effect on social motivation.
But wait, if these neurons represent the negative state of being alone, activating them shouldn't feel good, right?
Precisely. That's the other side of the coin. They tested this using something called a place preference assay. Basically, do mice prefer or avoid a place where something happened? And they found that the mice actively avoided the specific chamber, the location, where their MPN isolation neurons were being activated by the light.
So it felt bad. Aversive.
It strongly suggests that the activity of these neurons conveys a negative emotional experience, which likely contributes to that unpleasant, aversive feeling of loneliness we humans talk about.
So that feeling of unease or even anxiety when we're isolated, it might have a very real biological basis right there in the firing of these specific cells.
It certainly points in that direction.
Okay, so activating them feels bad and makes you want to connect. What happens if you do the opposite, turn them off?
Right. So they use another optogenetic trick to inhibit the MPN isolation neurons. They did this in mice that had been isolated.
So silencing the loneliness signal in lonely mice.
Exactly. And when they did that, they observed a reduction in the social rebound effect. The mice didn't seek out social interaction quite as strongly after isolation.
They also had less preference for socializing.
Yes. Also a decreased preference for social interaction compared to control mice. This demonstrates that these neurons aren't just correlated with social need, they were actually necessary for the full expression of that drive. You need them firing to feel that strong pull.
And this held up across different mice.
Yeah, they saw similar results when they specifically manipulated those MC4R expressing neurons, the key part of the isolation group, across both a high rebound FVB and the moderate rebound C57 strains.
Suggesting it's a pretty conserved function.
It seems to be, yes, consistent across different genetic backgrounds.
So these MPN isolation neurons are really looking like they're at the heart of this, driving the motivation to connect, and maybe creating that negative feeling of being alone.
That appears to be the case. Now, the next logical question is, How do these neurons exert their influence? Where do they send their signals to in the brain?
Right, the downstream targets.
Exactly. To understand the whole circuit, you need to know who they're talking to.
And where are these loneliness signals going?
They discovered projections, connections to a whole network of brain areas involved in really diverse functions. It's quite widespread.
Like what kind of areas?
Well, regions we know are involved in emotions. Places like the lateral septum, the bed nucleus of the stria terminalis, the BNST, which is often linked to anxiety processing, and also the habenula.
Okay, emotion centers. What else?
Also, areas involved in the stress response, like the paraventricular nucleus of the hypothalamus, the PVN. Regions involved in appetite regulation, like the arcuate nucleus, or ARC.
Appetite. Interesting.
Yeah, and even areas related to motor control, like the periaqueductal gray, or PEG.
Wow, that's a pretty extensive network. It touches on a lot of different functions. Let's maybe break that down a bit. They mentioned oxytocin, right? Often called the love hormone or bonding hormone.
Yes, that came up in the connection to the PVN. They observed an increase in the activity of specific oxytocin-producing neurons in the PVN. They call them PV and OX neurons during social isolation.
So being lonely activates oxytocin neurons. Isn't oxytocin usually associated with being social?
It's interesting, right? But here, it seems the lack of social contact triggers activity in these oxytocin cells. And when they directly manipulated these PV and OX neurons, activating or inhibiting them, it influenced the strength of the subsequent social rebound.
How so?
And furthermore, if they blocked the receptors for oxytocin systemically, that also reduced the social rebound.
So it suggests the oxytocin system is somehow involved in boosting the drive to reconnect after being alone.
That seems to be the interpretation. Feeling lonely might actually trigger the release of hormones that, perhaps paradoxically, make us want to be social again. It kind of makes intuitive sense, in a way, a drive to fix the deficit.
Okay. What about those brain regions you mentioned linked to negative emotions, like the habenula?
Right, the lateral habenula, or
Yeah.
LHB, they saw increased activation there during social isolation, and also in the lateral septum. As you said, these are areas known to be involved in processing negative emotional states, aversion, disappointment.
And did the MPN isolation neurons connect there?
Yes. And crucially, when they specifically stimulated only the projection, the pathway, from the MPN isolation neurons to the LHB,
What happened?
it caused the mice to avoid that area.
Just activating that one connection felt bad.
Exactly. It further supports the idea that this circuit contributes to the negative aversive quality of social isolation.
And you mentioned a connection to appetite regulation in the ARC, the arcuate nucleus. People often say loneliness affects their eating habits.
That's right. And the study found a potential neural basis for that. They saw increased activity in known appetite-suppressing neurons within the ARC during social isolation.
Which fits with what? People losing their appetite when lonely.
It aligns with observations, yeah, that isolated animals, and sometimes people, can experience weight loss or changes in appetite, and they went further.
How?
They directly stimulated the projection from the MPN isolation neurons to the ARC. It inhibited food intake.
Wow, so activating the loneliness pathway directly suppressed eating.
It suggests a direct neural link between social need and the regulation of hunger. It's almost as if the brain, when feeling socially deprived, might prioritize fixing that social need, perhaps even dampening other fundamental drives like hunger temporarily.
It's all painting a picture of a really complex, interconnected system controlling this fundamental need.
Okay, we've talked a lot about the isolation neurons. What about the other side, the MPN reunion neurons? How do they fit into making you feel socially satisfied?
Right, the satiety signal.
Yeah.
Well, a key finding was how they interact with the isolation neurons. The researchers found evidence suggesting that the MPN reunion neurons actually directly inhibit the MPN isolation neurons.
And the reunion neurons were mostly GABAergic, inhibitory. Exactly, makes perfect sense neurochemically. Social contact comes along, it activates these inhibitory reunion neurons, and they then put the brakes on the excitatory loneliness signal coming from the isolation neurons.
It's like a direct off switch.
A negative feedback loop, essentially. The need, isolation neurons firing, drives you to connect, and the connection, activating reunion neurons, then shuts off the need signal.
That's elegant. Did they test this functionally?
They did.
Yeah.
Using optogenetics again, they artificially activated the MPN reunion neurons in mice that had been isolated.
So turning on the social satisfaction signal in lonely mice.
Yes. and it led to a decrease in their subsequent social rebound. They sought less social contact afterwards.
Supporting their role in social satiation, making them feel like they'd already had enough social time.
Exactly. And interestingly, remember how activating the isolation neurons felt bad?
Yeah, the mice avoided it.
Well, activating these MPN reunion neurons conveyed a positive feeling. The mice showed a preference for the place where these neurons were stimulated.
So being around others isn't just stopping the negative feeling of loneliness, it's also actively generating a positive, rewarding sensation.
That appears to be the case,
Huh.
which also makes intuitive sense, right? Social connection generally feels good.
Definitely. Did they find connections to reward circuits?
Yes, they found that these MPN reunion neurons project to the ventral tegmental area, the VTA.
The VTA, a major hub in the brain's dopamine-based reward system.
Precisely. And consistent with that, they also observed an increase in the release of dopamine in the nucleus accumbens, the NAC, another crucial reward area, right when the mice were socially reunited.
So social interaction is inherently rewarding at a neurochemical level, involving dopamine.
It strongly suggests so. And dopamine seems important for the motivation part, too.
How did they show that?
They used a drug to block the reuptake of dopamine, specifically in the NAC. This effectively boosts dopamine signaling there.
And what did that do to social behavior?
It actually amplified the social rebound effect. Enhanced dopamine signaling made the mice seek out social connection even more strongly after isolation.
Indicating dopamine is involved in driving that motivation to socialize.
Yes, it seems to play a key role in the motivational aspect of social seeking.
Okay, wow. So we've covered the brain regions, the specific neuron types, their push-pull interaction, their connections to emotion, stress, appetite, reward, even key neurochemicals like oxytocin and dopamine. But there's still a basic question. How does the brain actually know? How does it detect whether we're alone or with others in the first place? What are the primary sensory cues driving this whole MPN system?
That's a fantastic question. And this part of the study yielded some surprising and I think really significant results. They started by testing the obvious candidates.
Like smell and hearing, especially for mice.
Exactly. But they found that simply being able to hear and smell other mice was not sufficient to satisfy this core social need.
Really? How did they test that?
They put mice in cages separated by a perforated divider. They could definitely smell and hear each other, but couldn't physically interact.
And
those mice still showed a substantial social rebound when finally allowed full contact. The sights and sounds and smells weren't enough to switch off that loneliness signal.
that is unexpected. We often think of those senses as being so important for social recognition and interaction.
They are important for aspects of social behavior, for sure, but apparently not for fulfilling this fundamental homeostatic need for social presence.
So if it's not primarily sound or smell, what else is there?
Well, they investigated two other possibilities.
Mhm.
Pheromone sensing, using the vomeronasal organ, which mice have.
Did that matter?
Nope. They found that mice, genetically engineered to lack the ability to sense these specific pheromones, still showed a normal social rebound. So it wasn't vomeronasal cues either.
Okay, so what's left?
Touch, mechanosensation. And this seemed to be the key.
How did they figure that out?
They used a pharmacological approach to temporarily interfere with the general activity of mechanosensory neurons, the neurons responsible for detecting touch, pressure, texture all over the body.
And when they blocked touch sensation?
It actually prolonged the social rebound effect. The mice stayed in that high drive state for longer, suggesting that physical touch is normally crucial for signaling social presence and satisfying the need.
Wow, so it's not just about being near others, it's about physically sensing their presence, likely through touch.
That's what the evidence strongly suggests. The profound takeaway here is that physical touch isn't just, a pleasant sensation. It seems to be a fundamental biological signal that directly tells our brain you are not alone, impacting these core need circuits.
Did they look at specific types of touch neurons?
They did. They used genetic techniques to eliminate or ablate specific known types of mechanosensory neurons in the skin.
Huh.
And they found that ablating certain types, like MrPV4 plus neurons or NaV1.8 plus neurons, led to a reduction in the social rebound.
Further emphasizing touch is critical.
Yes, confirming the vital role of touch sensation and how this social drive develops and gets resolved. Interestingly, the rebound wasn't completely eliminated by knocking out single types.
Suggesting multiple touch pathways might be involved?
It likely means that multiple types of touch receptors contribute to signaling social presence. It's probably not just one specific kind of social touch receptor.
This focus on touch, it reminds me of some classic psychology research, doesn't it? Like Harry Harlow's work.
Absolutely. The researchers explicitly drew that parallel. Harlow's famous experiments with infant monkeys choosing between a wire mother that provided food and a soft cloth mother that provided comfort.
The monkeys overwhelmingly preferred the cloth mother, right?
Yeah.
Showing that need for contact comfort.
Exactly. Inspired by that foundational work, these researchers designed a comfort touch preference assay, but for mice.
What did that involve?
They gave isolated mice a choice. They could spend time in a standard kind of hard plastic tunnel or in a tunnel lined with soft cloth.
And what did the isolated mice choose?
Unlike the mice that were group housed, who didn't show much preference, the isolated mice showed a strong preference for the soft cloth tunnel.
Seeking out that tactile comfort when feeling alone, that makes you think about, I don't know, why weighted blankets feel comforting maybe, even though it's not another person.
It's a powerful connection, isn't it? It really highlights an innate drive for that tactile comfort, especially, it seems, when socially isolated.
Okay, but did interacting with the cloth just feel nice, or did it actually impact their underlying social need? Did it reduce the rebound?
It did. That was the crucial experiment. They found that allowing isolated mice to interact with just the soft cloth tunnel with no other mice around actually reduced their subsequent social rebound when they were later put with other mice.
So the soft touch was partially fulfilling their social requirement, substituting for real social contact in a way.
It appears so, at least partially. And here's the neural link.
Yeah.
They even showed that interacting with this soft cloth inhibited those MPN isolation neurons
Turned down the loneliness signal,
and activated the MPN reunion neurons.
turned up the satisfaction signal.
Exactly. The soft touch was essentially mimicking the effects of actual physical social contact right down at the level of these key neurons in the hypothalamus.
So it really looks like touch isn't just, one way we interact socially among others, like sight or sound. It seems to be a really fundamental sensory signal to our brain about our social environment.
That's precisely what these findings strongly indicate. Touch appears to be the primary sensory modality, or at least a critical one, that informs the brain about social presence or absence and directly triggers these neural changes linked to social need and social satisfaction.
Well, this has truly been a deep dive. Absolutely fascinating stuff. Let's just quickly try and recap the main takeaways.
Yeah, sure. So we've seen the identification of these two key opposing neuronal groups in the hypothalamus, the MPN isolation and MPN reunion neurons.
Right, acting like a dynamic switch or a thermostat.
Exactly, regulating our social needs and the feeling of social satiation, moment by moment.
And this whole system seems to operate using similar principles to other absolutely essential homeostatic systems like hunger and thirst.
Which really emphasizes how fundamental, how evolutionarily deep this need for social connection must be. It's not just a nice to have.
Absolutely. And the critical, maybe even surprising, role of touch as the key sensory input that tells the brain about our social world and drives these neural changes.
Not just hearing or smelling, but actual physical sensation seems paramount for this particular system.
This new understanding, it feels like it has some pretty significant potential implications, doesn't it?
I think so, especially when you think about mental health issues linked to social isolation, like depression or anxiety, or even physical health problems, which are also associated with loneliness.
And maybe even for understanding disorders that directly affect social behavior, like autism spectrum disorders, perhaps.
Potentially. By getting a clearer picture of these fundamental brain mechanisms, the basic wiring, it might open up new avenues down the line, new ways to think about understanding or maybe even treating these conditions. It's still early days, of course, especially translating from mice.
Of course, but understanding the foundation is key.
Definitely.
So, as we wrap up this deep dive, I guess I'm left pondering something. What does this new understanding of just how fundamental our social needs are and the specific role of touch, what does it suggest about the impact of our increasingly digital world?
That's a big question.
Yeah, a world that's often less about physical presence and, more virtual. How might that reliance on virtual connection versus actual physical presence be influencing our own social homeostasis, our own internal thermostat?
It's a profound question to consider, for sure.
Yeah.
How does our biology interact with our modern environment?
Definitely something for all of us listening to think about. How do you experience your own social homeostasis? Well, thanks so much for guiding us through this fascinating research.
My pleasure. It's really exciting work.